Good day everyone and welcome back to our classes. Today we will discuss Protista. Protista na one of the kingdoms where all the organisms wey dey on this earth dey divided into. If you remember, we dey use the 5 kingdom method of classification wey Robert Whittaker bin design. And for inside this 5 kingdom method of classification, the second kingdom after Monera na Protista. So wetin dis protists, wetin dem be? Dem be unicellular eukaryotes, so na unicellular organisms, and dem dey eukaryotic in nature. We get video where we don discuss eukaryotes and prokaryotes for us to know the differences wey dey between all these things. So these protists na eukaryotes dem be, and they dey get, many of them get either cilia or flagella or pseudopodia. All these things na structures wey they dey use for movement, for their locomotion. This one dey very important. Now, all these protists, some of them dey heterotrophic, meaning say they dey chop, they dey look for materials to use as food. They go chop different materials, ingest those materials as their food. Or some protists dem dey autotrophic, meaning say they go generate their own food. They fit use sunlight or any other source of energy to generate their own food. Now, for this kingdom Protista, 4 major phylum bin dey. If you remember our method of classification, we don discuss this one before too. Hierarchical method of classification. After kingdom, the next thing na phylum. So the 4 different phylum wey dey for this kingdom Protista na Euglenophyta, Protozoa, Chrysophyta, and Pyrophyta. Na 4 major phylum be that wey dey, phyla wey dey for this kingdom. Now, these 4 different phylum, we no go fit discuss all the 4. So we go take out just 2. We go discuss the Protozoa and we go discuss the Euglenophyta. Now, the Protozoa, if you come for the phylum Protozoa, for that phylum, we get different members wey dey the phylum, such as amoeba. Amoeba na the most popular guy wey dey there, popular organism wey dey there. Apart from amoeba, we get paramecium, and we also get trypanosome. Now, wetin be some of the characteristics wey dis protozoa dem dey get? First, many of them dey microscopic, but na eukaryotes dem be, so na only microscope we go fit take see them. Now, we go begin talk about some of the characteristics wey protozoa get. Now, remember say I been talk say this protozoa, amoeba dey there, paramecium dey there, trypanosome dey there. Now, some of their characteristics na say dem be microscopic organisms, so we go fit only see them if we employ the use of the microscope. Many of them dey reproduce through binary fission. You know wetin binary fission be? Na division of parent cell to form 2 daughter cells. Dem dey unicellular. Many of them na they dey aquatic in nature, so their habitat where you go fit see them na for inside water, or area where water dey, or area wey dey damp. Any place wey dey very dry or water no plenty for there, you no go see them. Now, amoeba na protozoa, and amoeba get wetin we dey call pseudopodia. Na structure wey dey for the body of amoeba wey dey use for locomotion, for im movement. So as e dey move around with that pseudopodia. E dey important to know that amoeba get nucleus, and that nucleus dey carry all the genetic material. Remember say we don discuss the differences wey dey for prokaryotes and eukaryotes. Protists dem, like this amoeba, na eukaryotes dem be, so dem get nucleus wey dey well defined, and that nucleus dey carry many genetic materials. Now, amoeba get contractile vacuole. This contractile vacuole get we dey use contractile vacuole for osmoregulation, so e go dey regulate the amount of water or solute or salt wey dey inside the cell. This one dey important. Now, amoeba dey feed through engulfing. So many times, if you see material wey you wan ingest, wey you wan chop, e go flow, use the pseudopodia waka reach that place where the material dey, come engulf am, meaning say e go cover am. If e cover am, e go come form small membrane around that structure wey you wan feed on. So that small membrane, e go come form wetin we dey call food vacuole. This food vacuole na structure wey the cell don ingest, wey e wan chop, and if e form this membrane around it, e go use e get hydrolytic enzymes wey dey for the body of the amoeba. Those enzymes go come, they go deposit, the organism go deposit those enzymes for inside the food vacuole, so that amoeba go fit digest wetin dey inside that food vacuole. This one dey very very important. Now paramecium dey, trypanosome dey too. All these organisms dem dey under protozoa. If you look at the structure of paramecium, I go put the diagram up make you fit see am. You go see say paramecium get cilia. Dat cilia na little little thread like structures wey dey the body of the organism, and na for movement, na so that the organism go fit move. Paramecium dey get contractile vacuole, and as we don talk before, contractile vacuole na for osmoregulation. E go help the cell to control the amount of water or solute or solvent or salt wey dey for the body of the cell. If need dey to reduce the amount of water, the contractile vacuole go reduce, pour out some of the water wey dey inside the vacuole, push am out so that less water go dey inside the cell. And if need dey to increase the amount of water wey dey inside the cell, the vacuole go get bigger, retain more water for the cell. So that one dey the body of paramecium. Paramecium dey get cytoplasm too. Cytoplasm na gel-like substance. Na that substance wey be say all the metabolic activities and reactions for the cell, many of them dey take place inside that gel-like substance. So that one too dey. Food vacuole dey. Paramecium dey engulf, no, e no dey engulf o, e dey swallow food. So e go swallow food come digest that food. Paramecium dey get anal pore too. So that food, if you don digest am finish, take all the nutrients wey you want, the one wey be waste, the paramecium go push am out through its anal pore. Paramecium dey get cell mouth, e get pellicle. Pellicle na structure wey dey inside the paramecium, and this pellicle go dey help the paramecium with strength and support. Dey help strengthen the structure of the cell for the paramecium. Paramecium dey also get nucleus, and the nucleus dey divided into micro and macro nucleus. The major thing na say this nucleus dem dey carry the genetic material for the paramecium. So this one dey very important. Paramecium, trypanosome, amoeba, all of them dey under the phyla Protozoa. Now if we leave the phyla Protozoa, we will go to the phyla Euglenophyta. Now for this phyla, e get one important organism wey dey there. We dey call am Euglena viridis. Euglena viridis dey very important, and Euglena viridis are also microscopic unicellular organism. E get flagellum wey e dey use for movement. You know, flagellum resemble whip, so we dey call am whip-like structure wey Euglena dey use for im movement. Now Euglena get gullet, so food particles fit pass through that gullet and enter into the cell for digestion and for use. Euglena also get contractile vacuole, so and Euglena too, you dey find am for aquatic environment. E get contractile vacuole, so e dey fit regulate the amount of water wey dey enter and wey dey comot from the cell. This one dey very important. Now Euglena dey unique in that e fit carry out two processes. E fit carry out heterotrophic nutrition, and also e fit carry out autotrophic nutrition, meaning say e fit look for food particle, chop, digest am, use the nutrient for the various things wey e wan do, all the metabolic activities wey e dey carry out. E fit do that one, or if that one no dey, Euglena also possess chloroplast, and for that chloroplast, chlorophyll dey for inside that chloroplast, meaning say Euglena fit carry out photosynthesis. So if fit carry out photosynthesis, come even generate im own food, im own material in the form of starch, and e dey store those materials for special structures wey we dey call perenoids. So for these perenoids, starch molecules wey don dey formed by the Euglena organism, e go store am for those places, dey use am up whenever it needs this energy store. So this one dey very important. That one come make Euglena get wetin dey call eye spot, and this eye spot na special structure wey Euglena get, wey dey help Euglena with measuring light intensity, meaning say if the sunlight dey too bright and the organism no dey feel okay, with its eye spot, e go don realize say this intensity of the sun dey too much, and Euglena go find a way to comot from that place. But if that one no dey, then e go dey there dey enjoy himself. So and majorly, anytime wey light dey, like sunlight, most times Euglena dey switch to autotrophic nutrition, so e go dey carry out photosynthesis, dey do, produce im own starch for the materials wey e want. So this one dey very very important, and that sums up the major things about Uglena. Now we can't understand say this whole kingdom Protista dey, and we don't know the four filer wey dey under the kingdom Protista. We can't know the important one, Uglenophyta and Protozoa. As I dey leave this video now, I go put up different diagrams of all these organisms. Abeg, take your time, look through these diagrams, learn them, sabi how to draw them, practice them very well, so that if you reach inside exam, whether na JAMB or WAEC or anything, you will remember and you will fit put out all these things for the exam. Thank you for listening and have a wonderful day.